Hello everyone, welcome back to my channel. In this session, I'll be continuing from part 1 Kingdom Monera. The link will be provided in the description box. So let me continue with the part 2 Kingdom Monera, types of the bacteria. So before I continue with this session, do not forget to like, share, comment and subscribe. There are two types of bacteria majorly. The first one is Archaebacteria, the second one is Eubacteria. So today in this session, I will be explaining you detailedly about the type 1, that is Archaebacteria. Let us look into the characteristics of the Archaebacteria. Archaebacteria are commonly called as primitive prokaryotes. Why primitive? Primitive because these are tiny or very small organisms. These were believed to evolve soon after the first evolution of life. Archaebacteria is very importantly characterized by the absence of peptidoglycan, which is present in the cell wall. Here it is absent. So what is present here? Mainly the protein as well as non-cellulosic polysaccharides are present in the cell wall of the Archaebacteria. Archaebacteria live in such conditions which we call it as extremely hostile condition. This means very few organisms can dare to live in these conditions. Example, they can live in salt pans, salt marshes, hot sulfur springs, sea vents, etc. Archaebacteria are of two broad categories. The first one is obligate anaerobes. Anaerobes are those organisms which live without or in absence of oxygen. The second one is facultative anaerobes. Obligate anaerobes, here the Archaebacteria can live under anaerobic conditions only. That means they get killed in the presence of oxygen. Example, Methanogens. Second type of Archaebacteria is facultative anaerobes. These are the bacteria which can live under aerobic conditions, and also they can bear anaerobic conditions comfortably. For example, Halophiles, Thermoacidophiles. We have learned that under obligate anaerobes there is only one Archaebacteria, and that's very important Archaebacteria, which is called as Methanogens. Nutritionally, these are autotrophs. Very important point to be noted here. Methanogens lives in marshy areas. Why under marshy areas? Because they can convert formic acid and carbon dioxide to liberate methane with the help of hydrogen. On the screen you can see the stomach of the herbivore, that is cow. So here the Methanogens acts as a symbiotins. How it is symbiotins? Let us see. Methanobacterium lives inside the rumen or first chamber in the stomach of herbivore animals. It helps in digestion of cellulose and its breakdown into glucose, thus providing a source of energy for the cattle. This is done by the fermentation process. Methane gas is released in the atmosphere during exhalation. The next major category under Archaebacteria is facultative anaerobes. Under them, the first type of bacteria which we have learned is Halophiles, or it can be also called as Halophiles. Halophiles, by the name, these Archaebacteria live in extreme salty conditions or substrata. It's a place where they can survive, like salt pans, salt beds, and salty marshy areas, or we can also call it as salt marshes. Example, Halobacterium or Halococcus. Anyone you can write. Halophiles are aerobic chemoheterotrophs. For their nutrition, you have to look into the part 1 Kingdom Monera, where I have explained, and the link has been given in the description box. Halophiles cell membrane consists of a red carotenoid pigment. Why this pigment is very important inside a cell membrane. This is mainly because the pigment protects these halophiles bacteria from an harmful solar radiations. We have already learned that facultative anaerobes can equally live in aerobic conditions as well as anaerobic conditions. Under aerobic conditions, I've already explained. Under anaerobic conditions, how do they survive or subsist? They subsist with the help of ATP, which is already synthesized by the cell membrane, with the help of the pigment system solar radiations, because of which they can live comfortably in an anaerobic conditions. There might be question arising in your competitive examination like halophiles are able to live under high salt conditions due to, there might be certain options. So let us see how they can live under these certain conditions. The first point, presence of special lipids in the cell membrane. Second, occurrence of mucilage covering. Mucilage is nothing but normally the bacteria will be having an envelope or a gelatinous substance which is like an wall to them. Okay, so that's called as mucilage covering. Third is absence of sap vacuoles. This also I've explained in part 1 kingdom Monera. You can look back. And hence plasmolysis, which mainly help them to live in salt conditions. Fourthly, it is because of high internal salt content inside these halophiles bacteria. This is how these bacteria can survive under such harsh conditions. The second type of Archaebacteria under facultative anaerobes is Thermoacidophiles, or we also call it as Thermoacidophils. By the name thermoacido, these Archaebacteria can live in dual conditions, that is both conditions. In high temperature, they can tolerate higher temperature as well as they can tolerate high acidity. Oftenly, these Archaebacteria lives in very high conditions, like where the temperature is too high. For example, geothermal vents, hot sulfur springs, where the temperature may be above 80 degrees Celsius and the pH will be low as 2. The example that you can mention is thermoplasma or thermoproteus. Usually these are chemosynthetic autotrophs bacteria. A very important key point to be noted here. Under aerobic conditions, under presence of oxygen, they usually oxidize. Sulfur to sulfuric acid. Under anaerobic conditions or lack of oxygen, reduction takes place. That is, the sulfur can reduce to H2S. Some of the bicarbonates can also be precipitated under anaerobic conditions into the carbonate form by their activity. There might be question arising once again in your competitive examinations. How can these bacteria, that is thermoacidophiles, be able to handle both extreme condition, high temperature and higher acidity level? This is because, one, they have branched chain lipids, that is making their cell membrane very complex. Okay. Second one, they have special resistant enzymes which will not denature in the extreme conditions. That is very important. Special resistant enzymes which can survive even in the extreme or harsh conditions. Important note, Archaebacteria are also known as ancient living fossils because they represent one of the earliest forms of life which experimented on the absorption of solar radiations for the first time. Important uses. Archaebacteria are employed in the production of gobar gas from dung and sewage. In ruminants like cow and buffalo, they cause the fermentation of cellulose.